Welcome, everyone. Thanks for tuning in to the Moose Family Speaker Series on this beautiful day. First, some logistics. We do expect a large number of people who will be streaming in from all over, so as you've already started to do, use the chat to introduce yourself, and also use the chat for any questions that you have throughout today's webinar. Send the question to everyone using the drop-down bar directly above the chat box. And then to give ample time for discussion, today's webinar will run until 1:30. We have two speakers who will be speaking for a total of about 40 minutes. We've turned on closed captioning, and you can hide that by clicking on the live transcript button. My name is Carrie Jennings, and I'm the Research and Policy Director at Freshwater. We are a Minnesota nonprofit on a mission to inspire and empower people to value and preserve water. Since 1968, Freshwater has been a leading public nonprofit organization that uses science to engage people, from the region's residents to our lawmakers, on how to equitably preserve and improve water for future generations. It is our pleasure to partner with the University of Minnesota College of Biological Science to bring you this Moose Family Speaker Series. This lecture series was named after the late Malcolm Moose, who was the president of the University of Minnesota from 1967 to 1974. Freshwater and the College of Biological Sciences have partnered together for years to bring cutting-edge science on water topics to new audiences. Before I introduce today's speakers, please join me in thanking all of our generous sponsors for making this program possible. The Minnesota Board of Water and Soil Resources, improving and protecting Minnesota's water and soil resources by working in partnership with local organizations and private landowners. The Metropolitan Council, fostering efficient economic growth for a prosperous Twin Cities region. Minnesota Pollution Control Agency, a state agency committed to ensuring that every Minnesotan has healthy air, sustainable lands, clean water, and a better climate. And the Minnesota Department of Natural Resources, a state agency committed to creating a healthy, sustainable, and livable Minnesota. Now, if you are in the audience live, you could welcome me and join me in welcoming Drs. Genevieve Ali and Kamini Singha. Dr. Genevieve Ali is the Associate Professor in the Department of Geography and Department of Earth and Planetary Sciences at McGill University in Montreal. She studies surface and subsurface runoff processes, as well as water exchanges across the soil, plant, and atmosphere continuum in forested and agricultural watersheds. The main thrust of her research is water, sediment, nutrient, and biota connectivity in quasi-pristine as well as human-impacted landscapes. Dr. Ali is the former co-holder of a research chair in watershed systems. She also served as an invited expert to the United States Environmental Protection Agency Science Advisory Board and is a recipient of the Young Scientist Award of the Canadian Geophysical Union. She earned her Bachelor's of Science and PhD in Environmental Geography from the University of Montreal. Dr. Kamini Singha is a University Distinguished Professor and the Associate Dean of Earth and Society Programs at the Colorado School of Mines. Her research interests are hydrology and environmental geophysics. Dr. Singha is an award-winning teacher, a recipient of the U.S. National Science Foundation's Career Award and the Early Career Award from the Society of Environmental and Engineering Geophysics, a Geological Society of America Fellow, and a former Fulbright Scholar. She served as the U.S. National Groundwater Association's Darcy Lecturer in 2017 and was the AGU Witherspoon Lecturer in 2022. She earned her Bachelor of Science in Geophysics from the University of Connecticut and her PhD in Hydrogeology from Stanford. Drs. Ali and Singha, thank you for being here today. The screen is yours.
Great, thank you very much. So I'll just take a minute to share my screen and then I'll be ready to go. Okay, so you should be able to see my screen. So hi everyone, thanks a lot for having us. So I'll be starting off and then Kamini will be following me. And so yes, today we're talking about salt sponges and river livers. I'll be covering the salt sponge part of the talk. And so I guess I'll start with some context. We all know that how precious a resource water actually is. And while we all need water for subsistence, as echoed in the introductory comments that Carrie made, there are also a lot of water issues that we need to contend with. And so depending on where we live, we may be facing recurring instances of too much water, so floods, or too little water, so droughts. And then we could be facing recurring water contamination problems as well, eutrophication being one of them. And so eutrophication is a phenomenon of, I guess, overabundance of nutrients in water, which can turn our lake water or river waters green with algae. Now, I guess as a hydrologist, my main focus is on how water moves in any landscape. And there are a few different questions that my group and I focus on, but today we're going to focus on only two of those questions. So one of those questions is really, how does water get to a river or to a lake? And generally speaking, we know that it is via runoff, so larger movements of water either above or below the ground surface. But then we also have to ask the question of how come rainfall and snowfall are actually downward movements of water, whereas runoff is mostly lateral movement of water. So how is that transition from vertical to mostly lateral movement actually achieved? The answer to that question lies in the big part in the critical zone. And so the critical zone is the zone that basically starts at the top of the trees and that ends at the bottom of groundwater. And it is the zone within which, yes, we do have water, but we also have air, soil, rock, and living organisms that coexist and interact with one another. And so my focus today is really going to be on soil because it is a transition zone between the surface and the groundwater. And as a transition zone, it can really modulate not only how runoff is generated, but also how contaminants are actually exported from land to river. And so here I'm just showing you another schematic of the critical zone. And if we were zooming on a real soil profile, we would actually have something that looks like this, with different soil layers that have different thicknesses, different colors, different textures, different chemistries, etc. And so as we get rainwater and snowmelt water into the soil, that water can move vertically for a while, but often there will be something that will trigger a direction shift from mostly vertical to mostly horizontal. And of course, we can also have groundwater from below that rises into the soil profile per se. And so I really like to think about different soil layers as different sponges, which are superimposed onto one another. And so then what that means is that in any given location, the storage and the release of soil water and contaminants will basically depend on how many different sponges we have superimposed, what are their physical characteristics, how dry or wet each of them is, etc. So my group and I really look into those elements at individual locations, but we also try to look at variability in space in different types of systems. We do know that soil development is influenced by a bunch of things, including climate and vegetation. And so, of course, we can expect that soil sponges in different climates and in the different types of vegetation will also be different. The question is just different how. And so for the next few minutes, what I'm going to do is I'm going to take you on a very quick tour of, I guess, soil sponge behaviors in different landscapes that we've been studying across Canada, so at some of our Canadian study sites. And so what I'll do is that I'll start with soil sponges in riparian areas, so very close to river channels. And then I will continue with a forest example, and then I'll basically wrap things up with a quick word about an agricultural example as well. Okay, so my first example, I'm taking you to Manitoba. So that's the Canadian province that is located just north of North Dakota. The watershed that I'm focusing on today has a mix of forest and agricultural land use, but really when we studied it, our focus was only on near river areas, so riparian areas. And basically what we wanted to do there was to figure out when groundwater rose towards the ground surface. We also wanted to detect instances of water pooling above the soil surface near the river. So what we simply did was to build our own very shallow groundwater wells, and we installed them in the ground at a depth between 0 and 1.5 meters, so pretty shallow. And the idea really there was simply that if there were liquid water that was present in our shallow wells, that would be an indication that all of the holes in our soil sponges were actually filled with water. which would then give us an indication of what was actually happening below our feet. And so here, what I'm showing you is the data that we collected for 3 riparian areas in that watershed. So the pictures of what the riparian areas look like are in the top row there. And at the bottom, we basically created animated diagrams of our data just so that you could see how close to the ground surface the water table actually was, and how much water there was in the adjacent river channel as well. So that's why things are actually moving, because each frame in the animation corresponds to, I think, a different hour in time. And so the first thing you'll probably notice is that those 3 riparian areas behave in a completely different way, despite the fact that they're located relatively close to one another, and they pretty much receive the same amounts of snowfall and rainfall on an annual basis. If you were to focus on the riparian area on the right, you would see that the water table is typically not close to the ground surface, which is that brown line. And also that most of the time there's very little water in the stream channel per se. Now, if instead of looking at the riparian area on the right side, you shift your focus to the left side, you'll see there that the water table typically remains relatively close to the surface. And there were also times during the study period when the water level in the river channel was so high that the river was actually very close to spilling over its banks. Now, there are other analyses that we did and that I'm not showing here, and that provided additional evidence that shallow saltwater flow was generated more frequently in the riparian area on the left compared to the riparian area on the right. And so a conclusion for those riparian sites in that particular setting was that runoff generation was mostly in the subsurface, not overland, and it occurred mostly when the soil sponges were completely soaked with water, which is kind of intuitive and what we would expect, but as you'll see in my next 3 examples, that's not actually always the case. So my second example is from a completely different landscape. And so this time I'm taking you to a small, temperate, humid catchment that's located in Quebec, about 70 minutes away from where I am right now. And it is a beautiful forested region, as you can see on pictures here, with really, really strong seasonality. And so what that means is that we go each year from snow-covered soils and vegetation to green, lush vegetation in the summer, to really magnificent fall colors that are starting pretty early in our neck of the woods, I would say. Typically in late September, we start seeing those fall colors. And so the soils in that catchment are pot soils. You have a picture of those here, really strong or stark color contrast as you go deeper. And we have some rocks and gravels mixed in. One really key characteristic of that region is the fact that there's a soil layer that I labeled here with the arrow that is quasi-impermeable, very hard, very difficult for the water to get through it, and very difficult to even dig through, even using machinery. Now, we do have different colors and different textures for the different soil layers here, and so I guess you would see that the analogy of superimposed sponges for that site remains valid. That being said, the setup of superimposed sponges that you have on the right does not do a very good job in capturing a very peculiar feature of our real life site, and I'll talk about that in a minute. So on the right, you can see that all of the sponges are flat-sided. And so if you were in a situation where the pink sponge was very hard and did not let much water actually get through it, then here is what would actually happen. Water would infiltrate the soil, percolate from above vertically, and then when the water reaches the interface between the green and the pink sponges, the hydraulic conditions would actually change. So continuing to move vertically through the pink sponge would be too hard, and so instead the water would move laterally at the interface between the green and the pink sponges. At our Quebec site, so in real life, the interface between the 2 soil layers is not actually a smooth kind of flat-sided. It's not a flat line. What we rather have are hollows or depressions along that interface. And so in the case of water infiltrating the soil and then percolating from above, you would have water accumulating in that hollow there, fill it up before it spills over. And it's only when you have that spilling over occurring that you would have lateral soil water flow, so basically lateral subsurface runoff being triggered there. And so once we suspected that this may be happening in that watershed, we decided to look into it further. And so what we did was to make field measurements of the depth to the hard quasi-impermeable soil layer there. And then we produced a map out of it, which is what you have on the left here. And so this map did confirm to us that we had several subsurface depressions that can either be dry, or they can be in the process of filling up with water, or they can spill over in a cascading fashion, which would then trigger lateral soil water flow in that catchment. That was just a hunch at that point, and so we needed to provide field evidence or data to back that up. And so what we did then was to measure the height of water in those subsurface depressions on multiple occasions, just so that we could look at patterns of depressions filling up, spilling over, and generating soil water flow. And so on the map that you have on the right here, the red crosses correspond to dry or empty depression locations. Depressions that are in the process of filling up are in cyan, and then the depressions that are spilling over are in bright blue. And we did that on multiple occasions. And so in the animation that I'm playing here, you basically have each frame in the video that corresponds to different sampling dates or measurement dates. And one interesting observation that we made throughout that study was to realize that the north and the south portion of that small catchment were not actually spilling over at the same time. There was a difference in timing, and that difference in timing actually explained what we saw in terms of the chemistry of stream water at the bottom of that catchment. So we realized that there were some heavy metals that were present, not at the same time, in the stream water downstream, and same for some nutrients as well. And we could actually explain that difference in timing based on when we had the subsurface depression spilling over north or south of the stream. And so I just wanted to highlight an example to show that in some cases, we do not actually need the full soil profile. So all of your superimposed soil sponges to be soaked with water in order to trigger lateral soil water flow. In that particular example, what was really most important was the presence of an irregular interface between two particular soil layers or two soil sponges, and the ability of depressions along that interface to actually trigger lateral shallow subsurface flow, and therefore export of water and contaminants downstream. So I'm moving on quickly here, but I'm moving on to my third and last example, which this time is in agricultural system. So agricultural systems can be especially tricky to deal with because we need to understand how agricultural practices may modify not only the basic characteristics of our soil sponges, but also their hydrological behavior. And of course, we know that agriculture is highly dependent on water availability. On one end, we really do not want to be in a situation where you would have soils that are so dry that it could create stress for the crops that we're trying to grow on them. But on the other end, we also do not want to be in a situation like this where the conditions are so wet that we actually need a canoe to go from one end of the farm field to the other. And when we dealt with that particular situation on the right picture at one of our field sites a few years ago, we really had a lot of questions about what exactly the soil sponges were doing or maybe not doing properly underneath that farm field. And so going back to the sponge analogy, earlier when I was showing you that picture of my superimposed sponges, I did not actually comment on the fact that each sponge has various pore sizes that are present. So the pores are the holes that allow gases and liquids and tiny particles into the sponges, and they do exist in soils as well. So the smallest pores that we have in soils we tend to refer as micropores, whereas the largest ones we refer to as macropores. And macropores can be created by a bunch of different mechanisms. So macropores can be tunnels that are created by earthworms, or they could be tunnels that are left behind by dead roots, or they could be deep soil cracks as well that are caused by very dry conditions. So there would be desiccation cracks, or they could be cracks that are created by freeze-thaw cycles in very cold environments. And then, yeah, in the case of deep cracks in particular, something that's interesting about them is the fact that even when they seal off at the surface, they can actually remain open underground, which is something that hydrologists do care about quite a lot. And so you might be wondering why we care about macropores. We do care because they can really allow non-negligible amounts of water and contaminants to move very quickly below the soil surface, much quicker than the water and contaminants movement that we see in micropores, so the smaller pores. We can have macropores in any kind of landscape, not just agricultural ones, but we're particularly concerned about macropores in agricultural systems because it can allow water and contaminants to move very quickly from the surface to tile drains, for example, and then from tile drains to rivers and lakes, which could then exacerbate some existing water pollution issues. And then there's also research-wise a lot that we still do not know about the agricultural practices that may affect not only the presence, but also the density of macropores in agricultural soils. So would the use of organic amendments like manure, for example, affect the presence of macropores and macropore flow? What about the possible influence of cover crops that we're using as a soil health practice? Or what about the influence of heavy machinery? More studies or more research is actually needed about those elements. And so that's something that my group looks at as well. When water gets into soil, it can be pretty difficult to figure out whether it's moving in micropores or in macropores because it's translucent. And so that is why sometimes what we do is that we dye some water in blue, and then we use a rainfall simulator to make our blue water fall on the ground and infiltrate. And then after waiting a few days, we dig up holes in the soil to figure out where the blue water went by basically tracking the blue stains in the soil. And that can really help us better understand water movements in soils. And so here I'm just showing you one example of a blue dye staining experiment that we did in Manitoba a few years ago. We're only looking at 10 profiles there. We had 5 with liquid pig manure and then 5 without. And what we applied to each profile was enough blue water to simulate, I guess, to mimic a thunderstorm that would dump 32 millimeters of water on the farm over a 30-minute period, which is something that is not unusual in Manitoba in the summer. And so after a few days, we just went back, excavated the pots, and looked for the blue stains. And so we definitely found evidence of soil cracks that were there. So soil cracks formed in macropores. And there was some blue staining that was present around those macropores as well. Typically what we do is that we take pictures of the blue stain profiles, and then we delineate the blue stains on those pictures. So the diagrams that you have here just illustrate that there was a lot of variability in where the blue stains actually were, despite the fact that the pictures of the 10 profiles were located within just a few meters of one another, really. And then about 20 years ago or so, there were a group of researchers that developed an image processing algorithm to actually process those blue stain images to figure out what type of dominant soil water flow processes we have. So I'm not going to go through the details of the figures here. I'm just going to say that the darker blue colors represent soil water moving mostly through micropores, so the smallest pores, whereas the orange represents soil water moving mostly through macropores. And so in that particular study we did, we realized that macropore flow was present with and without manure, that soil water did not travel all the way down to the tile drains, which was good news, but soil water did travel deeper when manure was present, which was not so good news. And so in this example, soil water moved mostly vertically rather than laterally, and that was thanks to the macropores that were actually present there. Now, of course, there are other experiments that we could run, of course, and we have. So, for example, this past October, we did blue dye staining experiments for 12 soil profiles in Ontario this time with different soil conditions. So we're trying to assess the effects of cover crops and the effects of machinery-induced compaction on soil water flow through macropores. I just don't have results to show you yet because the analysis of the images is ongoing. And so in conclusion, I'll say that thinking of soils as sponges is really useful to understand how they can facilitate or not, the vertical and lateral movement of water, regardless of whether we are in riparian areas, forested areas, or agricultural areas. Within the critical zone, soils are key for modulating the rate at which water and contaminants move. But of course, there's a lot of variability in the way that they behave, not only in space, but also in time. Now, that being said, the soils are not the only components of the critical zone that can modulate or filter water and contaminants. There is a particular zone below rivers that can do that as well, which is exactly what Kamini is going to talk to you about. And so I'll just stop talking now and give her the floor.
All right. Thank you all so much. I'm happy to be here and take you a little bit down gradient from where we just were and talk a little bit about this idea of lateral flow and macropores, but we're going to move ourselves to the river environment. So I'm going to talk about what some hydrologists call a river's liver today and why you should care about it. So I think for most of us, we are very familiar with surface water systems. We're used to looking at streams. We can see contaminants coming into those streams. We can see indications of healthy stream ecosystems with fish and other biota. So most of us are really familiar with what's on the ground, but it turns out there's also just a ton of stuff happening underground that matters to us as humans as well. And this is a very complex slide showing a bunch of things that happen here, but the really key piece of this slide is that there are exchanges between the stream and the aquifer matter that surrounds it, and these provide a variety of what we sometimes call ecosystem services. And the idea of this sort of ecosystem service is that there are big gradients at this interface. And this interface is known as the hyporeic zone. It comes from two Greek roots, hypos meaning below and rios meaning flow. And at that interface, what we have is big gradients in dissolved oxygen and organic matter, as well as these water fluxes between surface water and groundwater. So groundwater typically has pretty low oxygen, low organic matter, and is high in solutes. It's also quite cool year-round compared to surface water, which varies quite a bit with whatever's happening in the air temperature. And so consequently, you've got this sort of important mixing zone here where groundwater can help provide, say, thermal buffering for fish refugia in streams as that sort of cooler water comes in. Surface water, meanwhile, has this really high oxygen environment, which mixes with this really microbial hotspot in the hyporeic zone to do what I'll call some biogeochemical work in just a moment. And that mixing provides some key ecosystem services. Some of them are things like metal uptake and mobilization in impacted stream environments. Others are nutrient processing to play on to what Genevieve was just talking about in a biogeochemically active subsurface. And most importantly, this hyporeic zone increases the residence time of waters in the stream itself. So rivers aren't just concrete canals, even though for some of us it sort of looks that way, but they really communicate with that material around them. And that is why the hyporeic zone has been called a river's liver, for its ability to sort of process material and do biogeochemical work. So how the hyporeic zone does this work happens because of these gradients. So we've got this low oxygenated environment in the groundwater compared to surface water that's exchanging. And what that means is we tend to see materials in the groundwater, which you would expect in lower oxygen environments, and materials that you would expect to see in higher oxygen environments up in the stream. And so what this means is that the hyporeic zone can process metals and nutrients like nitrogen, for example, where we might see nitrate in streams, and you see the oxygen there in the nitrate, right? We see that in the oxygen-rich environments, and it denitrifies as in these exchange places where we will see the reduction of nitrates to things like ammonia, as shown here, and eventually all the way to nitrogen gas in that exchange between surface water and groundwater. And we can also think about metals and how those change as a function of the differences in these two environments. But with respect to agricultural pollutants specifically and nitrogen load, as we move from land surfaces or regional groundwater into these rivers, if we don't account for the fact that the hyporeic zone has these chemical and biological transformations that it can do, it could lead to real errors in the way that we estimate what's happening with these materials. But it actually gets even more interesting, and that's that we see something completely unexpected. So in anaerobic or low oxygen environments, we have different expectations for what should be happening in the biogeochemistry. And that might be just as a function of mixing of groundwater or surface water, or here in this image, I'm showing you like a boulder that's sort of impeding flow. And our flow paths are going from being oxygen rich near the surface water to becoming less oxygen rich as they mix with that groundwater. But what's interesting is that we see anaerobic process signatures. So things that you would think would happen in low oxygen environments in high oxygen environments in these systems. So streams are typically bulk oxic, that area right near the surface water interface. So lots of oxygen there. And yet we see things like reduction of nitrogen, which we were just talking about, to say things like nitrous oxide, for example. That's an anaerobic process. And yet we see it in these oxic sediments. Now, this is super weird. It's kind of like if you did a candy survey of your sort of local town candy stores, and you found that there had been a run on Good and Plenty. Everybody knows that Snickers is a far superior candy product. So the only way that there would be a run on Good and Plenty is, in my mind, if we're out of Snickers. And yet, there are still Snickers in town. It turns out that what you're seeing here, though, is a local process very similar to what I'm talking about here. The reason there's a run on Good and Plenty is because a particular store may have sold out of Snickers, and now people are buying the next best thing on the shelf, and that's Good and Plenty. Same things happen in these environments, that microbes would way rather consume oxygen than they would denitrify things or reduce sulfate or reduce metals. But what we're finding is that in these bulk oxygenated systems, we have these less mobile, sort of pore scale anoxic zones. And what's happening is within this bulk oxic environment, these little tiny pore spaces that flow does not move through easily. And so what's happening is that you're getting anoxic microzones effectively within this big bulk oxic environment. And these can be really important, these little localized pockets, because they have really large residence times for water compared to the well-connected parts of the sediment. And so what that means is that they can really drive a lot of geochemical change within this bulk oxic space in a way that we wouldn't expect. And so that idea is something that lots of people are starting to think about here over the last few decades. And really in the last 10 years, people are trying to find ways to actually measure this. And so we've been looking at this as well. So there's a lot of open questions about hyporheic zone efficacy in terms of this biogeochemical work that we've just been talking about, in terms of how it controls the fate and the transport of different types of contaminants. We know that heterogeneity in space and in time are going to impact the hyporheic zone's ability to do work. But the complexity of this is something we're still trying to get our hands around. The image I'm showing you here on the left-hand side was made by Beth Hoagland as part of her postdoc work here at Mines. And she was really looking at the idea that heterogeneity in the conductivity of a stream to the aquifer around it can control fate and transport. She was specifically looking at metalloid transport in some acid mine impacted streams here in Colorado. But the idea holds for other materials as well. And what she found is that one of these stream systems, the one that was well connected between the stream itself and the aquifer surrounding it, and that's the image at the bottom, was able to partially self-remediate because of this ability for the hyporheic zone to process metals. But the other stream, which had was disconnected, had no such capacity. And so what's really interesting about the hyporheic zone is it provides a way for us to think about cleaning up materials in a natural way. Of course, this idea of connectivity and scale are complex, and we have big questions about how a changing climate might affect water quantity and quality in these systems, and thus the ability of groundwater surface water exchange to exchange and also to do work. But even without that future complexity, the factors that control the hydraulic gradients between groundwater and surface water, how heterogeneity matters, is really essential for us to understand these systems, and that in its own right is not easy. If we look inside of textbooks, we know that streams and their aquifers surrounding them communicate, and that streams might be losing water into aquifers or gaining water from the neighboring groundwater, just as a function of the relative elevations between streams and the groundwater around them, as shown on the left-hand side. And on the right-hand side, you see a system that's slightly more complex, where you can see gaining and losing sections of a stream in the same environment. And what this means is that predicting how effective the hyporheic zone might be difficult, because this idea of tracking flow paths can be really difficult. This complication between surface water and groundwater is complex, and it varies both in space and time. And these sort of textbook examples are way less complicated than what we know happens in the field. And here's a favorite image of mine from Kasahara and Wanzel. It's 20 years old now from a field site in Oregon. And what you're seeing in red here are a bunch of flow paths that they've outlined. And so these are hyporheic flow paths. They go from surface water into the groundwater system, back into surface water. And you can see that they differ over a variety of scales. And what I'm not telling you is, of course, this is happening in the third dimension that you can't see, this variability in scale. And that every time that there's an event, every time it rains, every time there's snow melt, the discharge in the stream changes, and that changes those flow paths too. And so what this means is that hyporheic processes change very dynamically in a way that makes them somewhat hard to predict, even though we know they're important. And it's way more than just the small headwater scale that I was showing you a second ago. I love this paper that Chicho Gomez-Velez published a few years back, where he was looking at the Missouri River watershed and the upper Mississippi and the Ohio. And what he's plotted here is exchange rates between sort of surface waters and groundwaters. And what you see here is that there are some, these are big watersheds we're talking about, and these red areas in these two systems show high vertical or lateral exchange. So we know that the exchange rate matters, but the other thing that matters is just the size of the exchange zone. So how big that hyporheic zone is, because that also, it's not just the movement of water and its flux, but also how big of a zone do you have to do biogeochemical work. So both of these things are really important when we start thinking about that river's liver, like how big is it and how fast can it process stuff. In terms of figuring out that size of zone, we have some characterization tools, but it's hard. Our options are pretty limited. A couple of things that we can do is, for instance, put in a series of piezometers. So what you see on the left-hand side here is a sort of famous image of a well field with tons of piezometers. And one might ask at some point if we've changed the system that we were initially trying to study by putting this many holes in it, sort of aquifer acupuncture. But from this, we can map out that extent of the hyporheic zone. The other option is for you and your friends to potentially get a big trash can and fill it full of dye or salt and dump it into a stream and see what happens down. And that's what I'm showing on the right-hand side. You see a concentration through time plot here. And in systems that are just advective, that was a canal, a concrete canal, you would see something like the black curve here. But in systems, in real river systems where we have that exchange, that transient storage of material in the subsurface, we see longer tails. Stuff is being retained for longer. That's that increase in residence time I was talking about. And that's that extra time that can be used to do that biogeochemical work. So we can track that, and with these concentration data, try to estimate the size of that exchange zone. But sometimes it's hard and it can be non-unique. So, sticking your head in the water also doesn't help. It turns out that we don't have great tools to look into the subsurface. But one of the things my group has been looking at, I'm trained as a geophysicist in part, is trying to use geophysical tools to answer some of these questions too. So going back to that same site that Kasahara and Wanzel published in that I showed you from 20 years ago that showed the red flow lines, we tried to sort of combine some ideas, introducing a dye into the subsurface. Actually, in this case, it was a salt, and then tracking it with some geophysical tools. So what I'm showing you here is some work that was led by Adam Ward, who was a PhD student who worked with Mike Gusev and I when we were faculty at Penn State. He's now a faculty member at Penn State. And what you see here are some cross sections through the earth. So you see the land surface here. We're looking down about 4 meters, and we're looking across 14 meters, very small streams. And what we're looking at is the flow at different rates. Now, before I go too far, what I'm showing you here in yellow is the extent, the size of the river at 3 different flows. And you're looking at a change in the electrical conductivity of the earth as we introduce something salty. So when we introduce salt into a stream, what's happening is, not surprisingly, the stream is getting saltier, but the background isn't really changing because the salt is maintaining its space in the stream. Now, you may be asking questions about why we work on streams this small, when I just showed you the data from Gomez-Velez that's looking at these really, really large scales. And part of the reason is because if we think about what that hyporheic zone is, that skin of material around a stream, It's everywhere. The Mississippi has a hyporheic zone, but the area of that hyporheic zone with respect to the area of the stream, the cross-sectional area, is really small. So it turns out that headwater streams, small streams, do a ton of work for us because that hyporheic zone is proportionally larger in these systems than it is in big river systems. It matters in both cases, but this is part of the reason why a lot of people spend time looking at small streams. And that is why we're looking at a small stream here. So what I'm going to do is I'm going to introduce the salt for a period of time. And what you're going to see is how the stream becomes saltier, but also what's happening around the stream. So the image here is a cross section again, 3 hours after we started injecting salt. Here is 6 hours, 12 hours, 24 hours, 48 hours after we've been introducing salt into the subsurface. In every single case, no matter the flow rate, what you see is that the stream itself gets saltier. Nothing surprising. But what's super cool is you can see how that area of yellow and green extends beyond the stream itself. What we're doing here is we're actually imaging the hyporheic zone for the very first time that anyone had tried to do this with a geophysical tool. And so now what we know is the extent of where that biogeochemical work could happen in the system. And what we're seeing is how it changes with flow rate in these cases. And in this system, although there's some contradictory evidence and other studies depending on the geology of a system, as your flow rate goes up, the hyporheic zone gets larger. Now, where this gets interesting is that we turn off the salt, and what you see is that this is 6 hours after the end of that injection of salt into the stream. These are low quantities of salt that are sort of ecologically safe. Just to point that out, this is like a little bit of salt compared to like even an average road salting event during the winter. But what you see, especially at the high flow scenario here, is that the stream itself has gone back to a background condition. You see at the 33 liters per second case, the stream itself is back red again, but you see this bathtub ring of salt that remains. There is your hyporheic zone. There is your increased residence time. There is the place in the system where you would expect metal uptake or nutrient uptake or processing to happen. And if we come back 24 hours later, we see that most of this salt is now gone, but not entirely. So if y'all were here at the Moose lecture last semester, then you met Pam Sullivan and Allison Anders, who defined sort of the idea of critical zone studies for this group. And one of the things that ties in with what they were talking about is that we think about the landscape in these integrated earth and environmental science spaces is either being sort of transformer or transporter systems. This is really important to a lot of the things that we're talking about. Does the material just move through a transporter, or is the material being changed in some way, as we were talking about denitrification, for example? And then that's where this connectivity of streams and scales really matters. Another big theme that in their work that plays on what Genevieve and I are talking about today is how the structure of the subsurface matters. It matters to the hydrology, and that consequently matters to the biogeochemistry. So when it comes to stream systems in particular, like we're talking about in critical zone studies, one way that we can talk about how effective the hydraulic or the hyporheic zone is in terms of processing materials can be through what are known as CQ or concentration discharge relationships. So this is another data set that we can use to think specifically about process. So what I mean by this is we're going to collect concentrations of various materials and compare them to the discharge of the stream. So these are some slides put together by my PhD student, Sarah Warricks, and I'm just going to walk you through three sort of simple examples of things that we might see in these CQ, these concentration discharge relationships. So I have a schematic on the left-hand side here that's showing you a plot of concentration and discharge. We do this in log space, and what you might see is a decrease in concentration with discharge. So what's happening here is your stream gets larger, the concentrations go down. Dilution. Like, right? More water, less concentration. And we see this happening in lots of systems where there's a solute source limitation, for example, and that we're sort of depleting that source and mixing in dilute water as the discharge goes up. So what's interesting about these sort of CQ relationships is they tell us about a system's sources and maybe its processing capability. Of course, we see exactly the opposite in some systems, which is that as discharge goes up, so does concentration. And this is what we think about as flushing of solutes, maybe from those soil layers, talking about some of the work Genevieve was just talking about. As discharge and these streams come up, flushing material into, and groundwater goes up, flushing material into these streams that lead to an increase in concentration. And we see this, for example, in metals quite a bit. And then, of course, there's systems where we don't see anything at all. The discharge in the stream changes, but the concentration basically stays the same. And this can happen when the reservoirs of solute that we're thinking about, whatever they might be, are reaching chemical equilibrium in a system really quickly because of, say, fast reaction rates, or that the flow that we're thinking about in this system is dominated by groundwater with really long residence times. Or maybe that we do have a flushing event, but the storage reservoirs have really high solute concentrations. So there's lots of ways that we can get these sorts of responses. And if we look across landscapes, and I really love this paper because it's from 300 different sites in France, in this case, where they're looking at concentration versus discharge in streams. And you can see a variety of different materials here. And down sort of the main diagonal, I've labeled some that follow what we were just talking about, this idea of mobilization, of dilution, of chemostasis. But then there's some other materials here that do not follow those sort of simple patterns that we're talking about. And they lead us to think about what else could be driving what's happening in these systems. So these plots like these, when we start to think about the concentration of various materials, can tell us about sources of stream water, but maybe more importantly, they can start to tell us about what is generating solute in the system, what are the bugs doing, what's the biogeochemistry or the transport that matters in these systems. So if we're looking at things like nitrogen or nutrient pollution more broadly, what is it that could be controlling transport and transformation, and maybe how that changes through season or through land use or through climate change. And the idea in the end is that maybe we can even make some numerical models that help us quantify this so that we can take our results from these very site-specific places that we work and try to scale out. And so these are the sorts of things that many people are trying to incorporate into their numerical models. So we can predict how much biogeochemical work a stream system can do with all of these changes that might be happening in terms of climate and land use, et cetera. And even simpler and smaller, how that system changes as a function of event and with season. And it turns out that we can also engineer these systems. So what I'm showing you here, some work is that was led by Skylar Herzog. He was a PhD student here at Mines. He's now at Oregon State Cascades, and his thesis was about sort of engineering the hyporeic zone. So what he did was introduce some impermeable barriers, which you see at the top, these little black triangles in a schematic he has there that were put in place to increase the exchange between the stream and the groundwater below it. So consequently increase the residence time of some flow paths that allow more time for that biogeochemical work. And the bottom image is a model that he made as he was sort of figuring this out before he actually implemented this in flume studies and then in some actual rivers. And you can see how that residence time changes. You get these really long flow paths between these barriers that he's put in. And so really, when we think about the processing of materials in the hyporeic zone, there's a trade-off here between the residence time of water, how long you can get stuff to be in the ground, and then the reaction timescales that you're interested in so that you can sort of try to optimize that in these systems. Of course, it's hard to optimize a natural stream system, but we can do it in these engineered systems, as we've shown here. So in conclusion, I guess what I wanted to wrap up with for all y'all is that, stream systems are complex. They're not that canal that some people that don't work in the earth and environmental system like many of us do, don't get to think about or don't get to see. And this idea of a river's liver, that hyporeic zone, is important for a variety of processes in terms of quantifying, predicting sort of biogeochemical work that can happen in these systems. And that little skin is really important to the natural remediation of nutrients. And with that, I will close.
Thank you so much. I think one of the most fun things about having you both on screen is seeing your reactions to each other and seeing you nod and agree and just, focus on some of these concepts. What motivated each of you to study this critical zone and these critical zone processes? Did you start out there? It sounds like you didn't.
Should I go first?
Yeah.
I can go first. I feel like when I started to study those kind of systems, I wasn't even thinking of the critical zone at all. I was really focused on just the soils and how water was moving through them and above them. And it kind of evolved naturally when I realized I couldn't focus just on the water. And so now I'm working with microbiologists like Kamini mentioned earlier, because and biologists and ecologists as well, because worms actually have a big impact on everything that I do. Same for plant physiologists, because depending on whether I'm focusing on trees or crop species, the plant water uptake is going to be completely different and influence my soil water movements as well. And so for me, that kind of happened organically. I didn't plan it, I guess.
Yeah, I guess my path is a little bit different. I fell into geophysics pretty early. I really liked math as a kid. I'm actually a fairly poor qualitative scientist. I'm not a very good descriptive scientist, and I liked math because it felt easier to like see things. But it turned out as I was moving into geophysics in the 90s, geophysics had a very different focus, and that was primarily on oil and gas, which was not what I wanted to work on. And I remember meeting a climate scientist from UBC, from the University of British Columbia, Gary Clark, who'd come to give a talk at my institution, and he had started as a geophysicist and had become a climate scientist. And so I asked him about that path because as a graduate student, it felt so far away. And he said that he wanted a piece of the earth to be his. And that really resonated with me as someone who was studying geophysics, because many times geophysicists are sort of making methodological developments. And I was less interested in that than thinking about the world sort of more holistically. And that moved me to hydrogeology. And then through that, I ended up realizing that hydrogeology is really complicated because of all of the ties to all these other pieces, and I feel incredibly grateful. I only know what I know, so a lot of the work that I do is in conjunction with, as Genevieve was just mentioning, with biologists and people that understand complexities of systems that I just can hardly get my hands around. But for me, it's been super fun to start to think about systems where I know nothing. You can ask the best questions when you don't know anything about it, or maybe I should say the worst questions, but like the worst questions, right? Because you're not hindered by the fact that you haven't been trained in that system. So for me, that's sort of how I ended up there.
That's great. And it sounds like both of you have worked on these unimpacted pieces of the earth and also very much human impacted. And what do you think the greatest impacts to the natural processes are by humans? Can you kind of sum it up, or is it just?
Yeah, I mean, I guess maybe I'll just say that simply as I'm thinking about this question, is that, man, humans we really really engage with our landscape in a really interesting way. Like land use change is a really big one. And then, when we think about the sheer amount of agricultural fertilizers that we use, obviously that's a huge load and a big change on a small environment. We see some different problems here in Colorado too, living in very arid states, right? We have a big water quantity issue out here, and there's a ton of people moving here. We're building houses that we don't have water for. Everything's on fire. So it's hard for me to even decide how we impact our environments most, but it seems like we cannot help but do it as humans.
Yeah, I would agree with that answer. And I would also say that sometimes the impacts are not as sudden or straightforward as we may anticipate. And so there are a lot of delayed effects. So it was interesting, Kamini was showing that diagram of chemostasis earlier. And then when you actually take that concentration discharge relationship dynamics and then apply it to the whole watershed as opposed to, let's say, just the aquaric zone, then there are different hypotheses as to why that might be going on. One of them being legacy of long-term agriculture. So basically nutrient stores that have historically built up to such an extent that it just takes a little bit of runoff for them to be mobilized because your system is transport limited, is not supply limited. The nutrients are basically ubiquitous. And as long as you have a little bit of runoff, they're going to be mobilized, which is why you have that flat line of the concentration is not changing regardless of the discharge values that you have. And that's something that kind of emerged only recently in terms of being integrated in the way we talk about agricultural systems, because in the past, we're kind of thinking that if there was something that we needed to fix, we could kind of set up a timeline in terms of number of years after mitigation approach that we should expect to see a result or a change. But if we're dealing with legacy sources, and that's something that I tell sometimes my students when we discuss in our group, there are some systems, agricultural systems, even if you were to stop fertilizer application today, it might take 50 years or 100 years for you to actually see tangible changes downstream because your historical nutrient buildups in soils are still there. So even if you stop contemporary sources, you still have those historical ones, right? And then it's the same kind of complicated answer when you think of climate change. I know that there's some areas here in Canada where, according to some climate change scenarios, we may see exactly or pretty much the same amount of precipitation over a year, but the distribution through the year will actually change quite a lot. And so we may go from a system that was snowmelt dominated to a system that is rainfall dominated. We might see more intense storms that are concentrated over very few months, which is going to lead to more water-related disasters because the dominant runoff generation processes are going to change as well, right? So it's a great question that you're asking. It's just that the answer is a very difficult one
Right.
to
Well, and
come
it
up with.
sounds like here in Minnesota, we do have money dedicated to kind of understanding and studying our water systems and our soils. And so we've done a lot of assessments on our rivers and lakes, and we know how they're impaired. Like, let's say, dissolved oxygen impairment. So we have streams with low dissolved oxygen. And, I think it sounds like from your talk, what we have to do is understand what the natural state is and what was going on in the soil surrounding the river and lake and the hyporheic zone in order to understand the natural state. I mean, we can't just introduce oxygen and expect it to return to normal, right?
Yeah, that idea of making sustainable systems and making streams do work for us has been an issue that people have been working at for a long time. In terms of how to engineer a system to make it more useful. And I do think it's difficult because the natural system is complex enough. I saw a question in the chat here even about like beaver dams and things, right? Like beaver dams, absolutely, they're the natural engineers of these systems and they enhance hyporheic exchange. So the natural systems are complex. And so then trying to figure out how to, I'm trying to find the word that is frequently used, and I've like blocked. There's a few words that like I find I struggle with, like sustainability is one, because it's like, what does that mean exactly, right? But when we talk about enhancing the efficacy of a system, there's a word for that too that I also find really difficult, because it means, what do you want out of the stream? What do you want it to do? And so how we perturb a system has consequences on the rest of the system. So I agree that this is a complicated piece.
So will a ditch in an agricultural landscape eventually develop some kind of normal hyporheic exchange?
Yeah, as long as so what happens sometimes, and it depends on what your system looks like. If you've got a lot of clay in a system, that's the one place where we see a very limited exchange in some of these agricultural systems. And so if there's clay lay, that will impede your exchange potentially between surface and groundwater. So that's a big one that we've had issues with. But if your soils are permeable and you've got water moving through some sort of ditch, it will exchange. And in fact, coming back to a water quality piece, and I know we've been talking a lot about quantity, and I can't help it because I'm in Colorado. We're really contingent on slop in the system. Like in all of these ditches that we have, we need that water leaking out of those ditches to even refill aquifers. And so the things we don't think about, like this idea of wasted water from some of these ditches, has a huge ecosystem benefit as well to groundwater. So yeah, yes, your ditch will exchange.
And I liked seeing some of your experiments, Genevieve, because they were in landscapes very much like in Minnesota. Southern Manitoba looks like northwestern Minnesota. And then the till landscape that you showed with the forest in Quebec looks like our morainic terrains in central part of the state. And so it seems like the healthier the soil, the better the connectedness of the pores, the more exchange you're going to have with the rivers. So a ditch in an agricultural landscape will behave better if it has soil porosity as well.
Yes, so I guess it depends, and I'm glad Kamini said what she's just said about the clay beds or the clay-rich soils underneath ditches, because in Manitoba, for example, in the Red River Valley, we have a lot of those, and therefore they're not as efficient as we may want them to be, regardless of whether it is to mitigate excess water when it comes to the quantity of it, but also the quality, as Kamini mentioned. And then there's also the fact that some roadside ditches or agricultural ditches are not left alone, right? So some of them are actually farmed, some of them are harvested, some of them need to be regraded. And so you were talking about natural systems and understanding them as much as possible before we perturb them. And sometimes it is difficult to find that quote unquote starting point, because when we're talking about sustainability or restoration, my question is always, we want to restore to what? And that's something that's very difficult. I was trained as a forest hydrologist. I only started working in agricultural systems when I got my first job in academia, basically. So at first, I remember my mindset was really, I'm going to quantify all those agricultural systems in terms of how they deviate from what forest systems actually do. Most of our hydrology textbooks are based on forested systems anyway, so those were my benchmarks. Except that forest systems are complex on their own and they don't actually behave in a single way, depending on whether you're dealing with mature forest, a forest that has been affected by wildfires before, insect infestations before. You're already not in the pristine state anymore, and so using them as benchmarks for restoration or sustainability goals is not something that's necessarily easy to do either, right? And so we have a phrase here. So in my university, McGill, so the strategic research plan has a phrase that has to do with sustainable working landscapes. So yes, the word sustainability is not super well defined, but at least there's the working landscape there to say that the goal is not to go back to the pristine forest from however many centuries ago, but to basically find some kind of balance between environmental sustainability and working landscapes. So it's just, we just need to figure out a way to quantify what that balance should actually look like for different types of landscapes. I think that we're all trying to get to that to some extent, but I wouldn't say that our community has a quote unquote streamlined way of getting there, right? We're just exploring and different subcomponents of the systems to see what may work or not work.
Right. And
does that feel like that makes sense, Kamini, or?
Yeah, and restoration was the word I was struggling with besides sustainability. Another word that you're just like, what do you mean to restore a system, right? So yeah, thanks for
right. That did come up in the questions. You know, is remeandering a straightened, widened stream good enough? Will that eventually help? You know, there are lots of stream restoration techniques that are applied to the landscape, and sounds like there's still a question. Let's move on to some of this. So we've been talking about kind of the transporter function of these critical zones. What about the transformer function, you know, with metals and nutrients? And I think one of the things I'm still struggling to understand is, you know, we have this excess nitrogen and phosphorus in the landscape, but we also have things of concern like mercury and sulfate. They impact the fish that people eat and the growth of certain crops that are harvested by Native Americans like wild rice. So, you know, understanding those exchanges seems really important in parts of Minnesota. Can you illuminate that at all for me? Is that too broad of a question? Grab on wherever you can.
No, it's I don't think it's too broad. It's definitely valid. I do tend to work on the quote unquote transporter function a little bit more when it comes to the transformer functions. I do look at wetland hydrology and biogeochemistry. And so, yeah, I see your point. And going back to something you were discussing with Kamini earlier, when we're getting rid of natural wetlands and trying to replace them with constructed wetlands, how do we do that if we're not entirely sure of how natural wetlands are actually doing that processing work beforehand, right? And so, and again, because there've been so many really neat advances in the past few decades, we now know, for example, that when it comes to wetlands and their transformer potential or ability, that larger wetlands are not necessarily the best. And most of the time, actually, the smaller they are, the better. And the more interconnected to one another they are as well, the better. But when we think of some of the constructive wetlands that we see in the landscape, we're typically not targeting those small ones, right? And so I guess that's where personally I've been really enjoying not just doing forest hydrology, which is what I've been trained to do, but kind of diving more and more into quote unquote agrohydrology as well. Because since it's a system that a few years ago I knew next to nothing about, I got to ask what I think are very naive and maybe a little bit dumb questions, but they actually opened a lot of doors for me. And they really forced me as well to I guess, work at the science policy interface and really try and understand what policymakers may be after and what landowners want to be able to achieve in a sustainable way as well, just so that I could reframe some of the questions that I'm interested in. So that's a very, I'm kind of going around your question on purpose. That's my long-winded way of saying that I think we're trying to get there more and more, and that's by not only bridging across disciplines within academia, but also bridging across from outside of academia as well, and then doing that more and more and finding that maybe some of my work may be more relevant because of that. And there's some of the questions that I never even thought I would investigate that I now am. So I'll stop there, I think, because I don't have any really specifics.
Well, if Genevieve is going to go around, then I'm going to go right out stage right. And that is because as a physical hydrologist, I have trouble parsing the response, the fate of particular contaminants. So my world is very physical in that I think that hydrology is the template under which, biogeochemistry lays on top. And so in terms of sulfate or mercury specifically, I can tell you exactly about, how water transports in the sort of area in which work can be done. But I'd actually need a biogeochemist to answer that for you in a meaningful way, because all of the nutrients respond so differently, metals respond so differently, metalloids respond differently than some other metals. And so I find biogeochemistry to be very overwhelming, just because to me, I don't know how they do it, right? There's like inputs, the magic of Gibbs free energy, and then outputs in these biogeochemical systems. And somehow my reactive transport colleagues see that landscape in a way that I will never be able to. For me, everything comes from first principles, and physics is easy. Biogeochemistry is really hard. So I don't think I can answer that question very well, just because I'm very naive to the differences between many of these different materials.
I agree. Physics is easy.
Yeah.
Chemistry, biogeochemistry, even worse.
And so you'll notice that both of us are kind of sticking to our area of expertise, regardless of whether it is the discipline or the type of landscape or landscape setting. And I think that and I'm not going to say just hydrologists, but geoscientists have really made a lot of progress in the past few years, is that we're acknowledging that we're very good at our small subcomponent, but we're really working together now to establish the connection. And so that connectivity that you were talking about, which is something that I'm doing so you were talking about the surface water, groundwater connectivity, and kind of looking at connectivity from a lateral and an upstream, downstream standpoint as well. But now we're more and more looking at connectivity of materials. So what kind of water connectivity do you need in order to get nitrate connectivity in your system, or phosphorus connectivity in your system, or mercury? And the answer will actually be different depending on the really biogeochemical peculiarities of individual chemical compounds, right? But that's something that's above my pay grade, I guess.
Yeah.
But I collaborate with people who allow me to do that and to really couple what they're doing with what I'm doing, which is really neat.
Yeah, I think people are happy to see examples in your data of like flushing of the system, and hear you say that specifically, that certain things might emerge that have been stored long term. And one of the problems we have in Minnesota and the metropolitan area is with chloride. We have lakes that are going to become too salty for aquatic life in 10 years based on the current trends. And will that flush out of the system? Can those systems be restored? I mean, maybe this is unanswerable and answerable too.
It's a great question, and it's this is a pervasive problem in the East. The sad answer is, I guess, that diffusion is a very slow process, and so a lot of these things are diffusing out of some of the things that both Genevieve and I were talking about, these sort of dead end pores. So we see contaminants in lots of systems, lots of different types of contaminants leaching out decades after that we've stopped applying them to some system. But of course, they mostly leach out at lower and lower concentrations. And so we coming back to that idea of how humans impact the environment, and Genevieve actually already said this, like, even if we stop doing everything we do right now, like, there will be these long-term tails, but at some point it will clean up. It'll just take time. And we've actually done some work. It was in Massachusetts, I think, looking at road salting as sort of a natural tracer. rather than introducing salts, which is what I was showing you in the example that I had here up in this sort of pristine headwater, we've used road salt as a tracer in some environments and some naturally occurring storms flushing that through. So we can see some of that. But yeah, that's a thing. It's a hard question to answer, you know, in terms of human safety in the case of road salt and then what it does to our environments. But yeah, I mean, it'll eventually go away, but it will take time.
That's good to hear, and something we should go ahead. Sorry.
There may be a way as well, and sometimes I was talking about the watershed-wide connectivity studies that we're doing in my group and with some collaborators as well. We feel like there may be a way for us to identify really critical nodes in the landscape. So, because yes, we know that things are transported downstream. So if we're going back to the transporter function, but they're not transported in a linear way. There are some particular nodes or critical points in the system that really act as gatekeepers. And if we could identify them, maybe we could establish or deploy mitigation strategies there in a really specific way to prevent them from allowing the downstream transport. That's something that we tried to do for the entire Lake Winnipeg watershed a few years ago, and we were able to identify, that's still theoretical, but we're able to identify 10% of the 1 million square kilometer watershed. 10% of it is a little more manageable than 1 million square kilometers. So 10% of the watershed that appears to have lots of local flooding on it on the years when the algal blooms in Lake Winnipeg are the worst. And so if it is more than just a statistical correlation, but rather an indication that those are really the gatekeepers that quote unquote open the gate in terms of transmitting water and transmitting phosphorus and nitrogen downstream, then if we wanted to do pilot studies and try and have an impact, then maybe those are the locations that we should target in priority. I feel like if we were able to do more and more of those studies, that would be beneficial, even just to test hypothesis and do pilot studies. The problem is that most often than not, the data is not actually available to do that. So in our particular case, we had to rely on remotely sensed data, and so our spatial resolution is relatively coarse. It would be great if we had 30 meter by 30 meter surface water and addition data to actually do that, but in order to do that with such a fine spatial resolution, then we need to really downscale and focus on smaller watersheds. So there may be a way, it's just data requirements or data availability could be an issue.
Well, we will try to temper the expectations of our lawmakers. They've invested now for about 30 years in monitoring and starting to clean up water, and they've set a date for when they want it done by, which is in the not too distant future. So I think we need to have a conversation about how long, what our expectations should be. But is it possible that by restoring the physical properties and the transporting aspects of these systems, the soil and the stream hyporheic zone, that we can expect the chemistry to follow? Do you think, if we just focused on the physical at this point?
I mean, it's a good question. I each system is so hard, and I'm trying to make big lumper assumptions here. And as a field scientist, it's a lot easier to be a splitter than a lumper. I feel like the thing about policy that makes it so hard is that you really need to be a lumper to be a policy person. And so much of policy is so hard because, the science is so piecemeal for the decisions that we have to make around things. Certainly starting with the physical template seems like a perfectly reasonable place to start. Right? It's a big help to start with that piece in my mind. The ways that we can think about building systems that increase residence times of water so that natural systems can do some work is a thing. And maybe that's enough in some systems and maybe it's just not in others. But I feel for policymakers trying to figure out how to make decisions on the data that we as scientists and our ivory towers sometimes, we're like, oh, but this detail really matters. And I don't know, maybe some of those details don't matter in the big picture of the thing, but it's definitely a difficult problem. So anyway, long answer short, I think that starting with the physical system at least has some legs.
But
Genevieve?
Then the question goes back to which quote unquote material movement are we trying to prevent, impede, or enhance, right? And so I remember one time I was giving a talk to an audience in which there were a lot of ecologists, and I was talking about water, chemical, and sediment connectivity in particular. And then I was basically discussing the fact that in most systems, we actually want to decrease water connectivity to prevent downstream flooding, but at the same time, hydropower generation companies don't like hearing that, and some ecologists either, because they want environmental flow requirements downstream to be satisfied, and you need water downstream in order for hydropower generation downstream to be sustained. You want chemical disconnectivity because you want to prevent contaminants from traveling all the way downstream. But then I had ecologists sit their hand and say, well, unless we're talking about invasive species, what I actually want is not ecological disconnectivity. I want ecological connectivity being enhanced so that I can maintain or enhance biodiversity in some portions of my landscape. And so then the question goes back to, are we focused on quote unquote chemistry or biogeochemistry only, in which case the answer might be relatively straightforward, or are we trying to look at multi-material movement and transformation, in which case what works for one is actually the total opposite of what may actually work or would be beneficial for the other one. And that's where trade-offs are really important, which makes me echo what Kamini just said, which is that I don't think I would be a good policy person, because it's very difficult to figure out what those trade-offs should be.
Yeah, well, you've got some people on. We still have quite a few people who haven't yet gone outside to the 75 beautiful day, but you do have some policymakers still on. You do have some scientists still on, and we need them to talk more to each other. I think there's been a success recently in Minnesota in that we have two new programs that have been established. One is to address soil health questions. The other is to address water storage. And we feel like those two things together can start to repair critical zone function across the landscape. What would your favorite practices? People are asking about specific plants or just cautions. We're about to go outside and till our gardens probably, or farmers are going to start planting oats and other things. Suggestions and cautions for sensitivity to our critical zone function.
So can I say that it's very environment dependent? And so there was a lot, so I guess this is international research, that's on Canada's side only, but there was a lot of research about conventional tillage versus no tillage, for example. There was a really big push towards no-till in Canada. I'm not quite sure how it went in the US. And so then we realized that yes, no-till has its benefits, but because of the crop residues that are left on the ground surface, we may in some instances actually have more nutrient mobilization because of the no-till. And that's something that in some systems was not anticipated at all, right? And so that's why I'm a little bit reluctant to, as Kamini alluded to, kind of lump things, because I think that it's really system dependent. And again, I think we're doing a better job now at trying not to look at a single landscape or landscape setting and then basically generalize to everything. Like for example, here we have a big new project that we're starting this year, where we're actually going to compare agricultural systems in the Great Plains, so in Manitoba, to what we have in Ontario. So slightly different soils, different climates as well, because we have more cold days with frozen soils, etc. And so basically testing different agricultural practices to see whether we get the same type of benefits, but using experimental setups that are really similar, so that we know that we're removing any kind of potential methodological bias and really just trying to compare apples to apples as much as possible. And there are more and more efforts in North America and elsewhere to do those kind of studies, right? So hopefully within a few years, we may be able to generalize things a little bit more, or say that we really need to stick to, I don't know, ecoregions or physical regions or hydrological landscape regions in the US, who knows, as a way to lump things up, if that makes sense.
I'm going to let the expert have the last word on that, since she knows a whole lot more about soil health than I do.
Well, I think maybe if you could find another way to sum up what you think the best actions would be for people, or maybe just the connectivity of the system. That I think you mentioned early on that we manage land in a kind of heavy-handed way, and that does impact water. So I don't mean to lead you, but just we have just probably about 8 minutes left, and I'm kind of looking for some type of summary or just next steps.
Oh man, you don't want.
We'll keep track of your research efforts and the work in the critical zone. But
Gosh, I'm like the worst person to ask those things of, because I feel like my summary for all these things is always like, it's hard. I think it's complicated. These systems are complicated, and how do we simplify any system we're working into its sort of base descriptor so that we can move forward is always a trick. And I don't think there's an easy way to make a broad generalization for a whole series of sites, because what controls the dynamics of those systems changes so much from place to place. That it's really difficult, I guess, from this perspective to say like, we should be engineering changes in all of these systems, because I don't think that's the truth. I mean, there's so many systems that, coming back to, I guess, just what I was talking about, that will naturally process materials, but then as soon as clays are involved, they won't. So then maybe you need to engineer that system. And so like, I do think that it's hard to think about broad brush solutions, which is what makes all of our jobs so difficult, is that every system is a little different. And the idea of numerical models as a way to sort of do what if scenarios, to me, is a really powerful tool. But of course, they also, that is the sort of broad brush way of looking at a system. But then you have to ask yourself in those models, is it okay that I ignored these complexities of my system in order to like move forward? And sometimes the answer is yes, and sometimes it's not. Like I was making assumptions that nitrate was conservative in some model I was making, to the horror of my biogeochemical colleagues, because I just needed it to move in the system and come up with something simple. And so is nitrate conservative? Of course not. Similarly, they were talking about one-dimensional flow. Nothing's one-dimensional, but they made those assumptions as biogeochemists because they needed to move forward. And so I think that the question is like, what are the things that you are willing to give up in your system to move forward? Is just a complicated question that's hard for me to answer. So I'm punting on this awesome question, which is like, I just don't know how to give something super clever that's gonna.
Well, no, I think you're saying scientists need to keep talking to scientists. Scientists need to translate to policymakers what they understand, and that we need to be sensitive to the river's liver and the soil sponge and those primary functions.
And I think that Kamini was making a good point earlier about what do we need to give up. I think that this is something we're all really in the weeds of our system, and so we need to focus sometimes on nitty-gritty details. But there's one hydrologist in particular, Hilary McMillan. She's really into perceptual models of catchments right now. And I find it very difficult to do, but basically you need to be able to summarize the functioning of your catchment through a diagram, which forces you to get rid of everything that you think is maybe just anecdotal or nitty-gritty, and really focus on the bare bones. And she's basically collecting perceptual models of catchments worldwide right now to be able to build a database and maybe learn from them. And so I think that initiatives like this one, the more we have of initiatives like this one, the more comfortable we will become at lumping things and then providing generalizations that are still physically based, right? And that we don't feel too uncomfortable actually making. So we're getting there, but personally, I'm not there yet either, right? So.
Okay, well, I think we are going to wrap up and give people their afternoons back. And thank you so much, Dr. Ali and Dr. Singha. We really appreciate your sharing your insights. And viewers, you can continue to submit questions to the chat, and we can try to follow up with the speakers. We will also provide, freshwater contact information. We can try to email you answers as we find them. But this concludes today's program. If you enjoyed it, please visit the Freshwater website at freshwater.org. Help us continue to bring public lectures like this to you and new audiences by donating or offering suggestions for topics and speakers. Thank you again, Dr. Genevieve Ali and Dr. Kamini Singha, and to all of you viewing us online for your participation. This lecture will be available to stream from our website, and a link will be emailed to you and all who have registered in the next day or two. Enjoy the rest of this beautiful day. Bye, everybody.